Scientists are one step closer to making artificial cells with lifelike activity thanks to groundbreaking research utilizing bacteria. Scientists have used bacteria to help develop advanced synthetic cells that imitate real-life capability. From bioengineering to life's origins inquiry, previous attempts to model protocells using microcapsules failed. So the researchers resorted to bacteria to construct complicated synthetic cells utilizing a live material assembly process. Professor Stephen Mann from the University of Bristol's School of Chemistry and the Max Planck Bristol Center for Minimal Biology, along with colleagues Drs. Kanzu, Nicholas Martin, and Mei Lai from the Bristol Center for Proto-Life Research, demonstrated a method for building highly complex protocells using viscous microdroplets filled with living bacteria as a microscopic building site. The team began by exposing the empty droplets to two types of bacteria. One population was collected spontaneously within the droplets, while the other was trapped at the droplet surface. The released cellular components were then trapped inside or on the surface of the droplets to construct membrane-coated bacteriogenic protocells comprising hundreds of biological molecules, parts, and machinery. The researchers discovered that the protocells could manufacture energy-rich molecules through glycolysis and RNA and proteins through in vitro gene expression, demonstrating that the hereditary bacterial components remain active in the synthetic cells. To put this technique to the test, the researchers used a series of chemical processes to physically and morphologically alter the bacteriogenic protocells. The bacterial DNA was condensed into a single nucleus-like structure, and the inside of the droplet was infiltrated with a cytoskeletal-like network of protein filaments and membrane-bounded water vacuoles. The researchers put living bacteria inside protocells to establish self-sustaining AP synthesis and long-term energization for glycolysis, gene expression, and cytoskeletal assembly as a first step toward the creation of a synthetic, living cell entity. Surprisingly, due to on-site bacterial metabolism and growth, the proto-living constructions developed an amoeba-like exterior morphology, resulting in a cellular bionic system with integrated life-like features. Achieving great organizational and functional complexity in synthetic cells is difficult, especially under close to equilibrium conditions, stated corresponding author Professor Stephen Mann. Hopefully, our current bacteriogenic technique will contribute to increasing the complexity of current protocell models, facilitating the integration of several biological components, and allowing the construction of electrified cytomimetic systems. Our living material assembly approach gives a possibility for the bottom-up production of symbiotic living, synthetic cell structures, said first author Dr. Kanzu, research associate at the University of Bristol. For example, employing modified bacteria, complicated modules for development in diagnostic and therapeutic fields of synthetic biology, as well as biomanufacturing and biotechnology in general, should be possible. Thanks for watching.